Hi guys, Dr. Harmon. Okay, in this video we're going to be talking about proteins. So let me share my screen so we can see the proteins. La, la, la. Okay, so we're looking at proteins. And the important thing to remember when it comes to proteins is that proteins are kind of the building blocks. These are the main Legos that make you. Okay? So just like with everything else, we have a monomer, and we start joining the monomers together into longer and longer chains until we come up with our complex macromolecule. So in this case, our monomer is what we call an amino acid. That's our single unit. And when we start joining amino acids together, we join a few together, and the bond that we have between these two amino acids is a peptide bond. So we have our peptide. And when we join quite a few of them together into a nice little chain, we call this a polypeptide. Poly for many, peptide because we have multiple peptide bonds. And then when we take the polypeptide chain and we get it long enough, we fold it, and sometimes we even have more than one chain. We fold them together to form our protein, which is the large macromolecule. The bonds, we use the same process we've been using, dehydration synthesis, dehydration reaction, to join these molecules together. We use hydrolysis to break them apart, but this time we call these bonds peptide bonds. Okay, so when we take our protein, there are molecules consisting of polypeptides. Remember, polypeptides are going to be polymers of our amino acids. Polymers, we're just putting several of the monomers together to form the polymer. And then we fold them into a three-dimensional shape. That three-dimensional shape is absolutely critical when it comes to proteins. And I'm going to say this over and over and over throughout this course. When it comes to proteins, shape determines function. Shape determines function. If we change the shape, we change the function. Most of the time when we say when we change the shape, we change the function, what that means is it doesn't function properly anymore. Okay, so proteins have carbon, hydrogen, oxygen, nitrogen, and some of them will have sulfur. Not all of them, but some. So previously, our carbohydrates and our lipids were just CHO, carbon, hydrogen, oxygen, for the most part. Our key elements for our proteins is CHON, carbon, hydrogen, oxygen, and nitrogen. And then, like I said, some of them will bring in sulfur, some do not. Shape determines function. I can't say that too many times because that is probably the most important piece of information you need to understand when it comes to proteins. Shape determines function. Okay, so when we're talking about our amino acids, and remember those are our monomers. Hang on, I got to back up because I oopsed and went too far and too fast. Okay, so monomers, these are amino acids or molecules that have an amino group and a carboxyl group. So we're taking our base hydrocarbon skeleton. We're going to put an amino group, that's one of our functional groups, on one side. We're going to put our carboxyl group on the other side of it. And then you'll notice there's a piece that says R. We call that a variable side chain. That R is just representing a fill in the blank here space. There are 20 different amino acids, and what makes those amino acids different is that variable side chain, that R group. So, for example, here at the bottom, you see we have alanine on the left, and the R group or side chain is CH3. On the right, we have glycine, and notice that the R group is simply a hydrogen. So the different R groups are what make our 20 different amino acids. Are you going to have to memorize those amino acids? Absolutely not. Depending on what you study further on in your school career, that might be the case. But for this course, no, you do not have to memorize the different amino acids. You will have access to a chart that goes through all the different amino acids whenever you need it. Okay. So each amino acid, and we use AA typically to abbreviate, has a unique side chain. The unique aspects of the amino acid, what makes that amino acid specific, what gives it its physical and chemical properties, really and honestly, are all about that side chain, because they all have the same base structure. So what's different is the side chain, and so therefore their chemical behaviors and their physical structural behaviors are different because of that unique side chain, that R group. Typically we can actually group the side chains, so we can say, okay, we have this type, and this type. So there are nonpolar or hydrophobic side chains, which means some of our amino acids are going to have nonpolar or hydrophobic behaviors. There are polar or hydrophilic side chains, giving that same characteristic, kind of like water. We have charged or ionic side chains. These are also going to be hydrophilic, so they will also react with water. The side chains will interact, and this is actually what determines the shape and therefore the function of the protein. So I always think of it kind of as a popularity contest. Some people like to really be around other people. Some people want to be as far away from other people, and some people really don't care. Well, there are going to be some of these side chains that want to be as far away from each other as possible, or they need to be protected from water, or things like that. And so when this thing folds, when the protein, the polypeptide chains, the protein folds, they're going to be on the inside protected. They're going to be away from these other polar molecules. The ones that are polar tend to be further outside. So you're going to find that the different things will attract and repel from each other, and this is what gives it that three-dimensional shape. So, formation of peptide bonds. To form a peptide bond, the carboxyl group of one amino acid, that's that COOH, is going to line up against the amino group of another one, that's the NH2. We're going to take the OH off of the one, and we're going to take one of the hydrogens off to release our water. Those two ends become sticky, and so they're going to stick together. And we do this over and over and over and over. This is what forms our peptide bond. And the more we bond together with our dehydration reaction, the longer the chain's going to be. Okay, so we take many amino acids, link them by those peptide bonds. Each polypeptide is going to have a unique sequence of amino acids and directionality. So we're not going to put the exact same amino acids in the exact same order over and over and over again, because that would be the same as writing the exact same letters for the exact same sentence over and over again. We've learned that if we mix up the letters, we can make all sorts of different words and sentences. Well, if we mix up the order of the amino acids, we can make all sorts of different structural molecules. Each polypeptide, each end is going to be chemically unique. One end is a free amino group. We call that the N-terminus. One end will always be a free carboxyl group. That's the C-terminus. Okay. So the sequence of the amino acids is what determines the shape based on how they interact with each other. And remember, shape determines function over and over and over. I told you that's going to come up a lot because it is the most important piece of information when it comes to proteins. When a polypeptide twists and folds, and remember that's because of how those different R groups interact, it's going to form our protein. So quick, I want you to think, pair, and share. How is the unique sequence of amino acids determined for a polypeptide? How do we know what amino acids go in what orders? That's an interesting question. We'll see if you remember. Okay. And the answer is the genes. Your DNA, your genetic information actually determines the order of the amino acids. And we're going to go into that in lots of detail as we progress through this course, just in case you don't remember. But the genes determine the amino acid sequences. Okay, so function of proteins. What do they do? Why do we care? And it's amazing how many people are going to say, oh, proteins give you energy. Guess what? Carbohydrates give you energy. Lipids give you energy. Carbohydrates are quick energy. Lipids are like long-term stored energy. Take more effort to break them down, but proteins are not really about energy. Yes, ultimately, in worst case scenario, if there's nothing else available, yes, they'll give you energy. But that's not their main function. That's not their main purpose. Okay, so functions include antibodies to help protect you from diseases, enzymes that carry out all the different chemical reactions or assist in creating new molecules. There are messengers that actually transmit signals. So, for example, some of your hormones would be protein-based messengers. We know other hormones are steroid or lipid-based, right? But some of your hormones are protein-based. They are structural. They provide structure and support. These are the key building blocks that you are made out of. They are used for transport and storage, so they can bind to and they can carry small atoms and molecules throughout the body. So when it comes to proteins, and we were talking about how they fold or how they build together, there's actually levels. There's a certain order in which they develop. So we're going to start with the primary structure. The primary structure is a linear chain of the amino acids. So here we have glycine, proline, threonine, glycine, threonine, glycine, glutamine, serine. Yes, I've dealt with these enough. I actually know most of the names based on their abbreviations. And you may learn to be familiar with some of them, but you can always look at the list. These are determined via the genes. The genetic material actually determines that specific order. It dictates then the secondary and tertiary forms as we go along, because it's all going to be based on the R groups and how they interact based on that initial chain. So our secondary structure, after we take and put the chain together, our secondary structure, they're either going to coil up into like, looks like a little slinky, or they're going to fold, kind of like, you know, when you were a kid and you made those little fans where you fold the paper back and forth and back and forth and back and forth. They fold into these little sheets. We actually call them pleated sheets or beta pleated sheets, but they form into either sheets or coils. That's the secondary structure. That's due to hydrogen bonding within the polypeptide backbone. Okay. So hydrogen bonding is what's going to determine if they fold like a fan or coil like a slinky. So our beta pleated sheet hydrobonds are going to be laying side by side. If we have our alpha helix, the slinky shape, the hydrogen bonds are between every fourth amino acid. That's what makes them actually coil up. Okay. So we've got our primary and our secondary. So now we're going to move on to our tertiary structure. Tertiary is the 3D folding, three-dimensional folding, due to the interactions between those side chains of amino acids. Okay. It's reinforced by the whole hydrophobic interaction, and then that we form what we call disulfide bridges between some of those side chains. Remember we said that the sulfur was going to come in sometimes? This is the place where sulfur is going to come in. That's what helps hold it into that specific shape. The covalent bond formed between the sulfur atoms of two cysteines are what makes that disulfide bridge. So they're like little links to help hold it into its shape. For some of the proteins, tertiary is the final structure, and it will just function as it is. Sometimes we will move on to one more level, which we call the quaternary structure. And in the quaternary structure, we actually take two or more separate polypeptides. And so you'll notice in the diagram, we have a paler color and a darker colored one. And they are going to get folded and mixed together. Only some proteins will have this quaternary structure where we're taking multiple polypeptides and putting them together. All four levels of the protein structure will determine the function because all four levels work together to determine the shape. And we all remember, shape determines function. Okay, practice. Okay, so we got a little real world scenario here. Cystic fibrosis is a serious, life-threatening disease that affects the lungs and digestive system of affected individuals. This disease results from the deletion of 3 nucleotides on the cystic fibrosis transmembrane conductance regulator, CFTR gene. So we're actually taking 3 pieces out of the gene. The gene codes for a transmembrane protein that regulates ion channels in epithelial tissue. So there's a specific protein that's going to control how certain molecules can move in and out of a cell. If we're missing those nucleotides, that's going to change the shape, therefore the function, right? So it says provide an explanation for how this deletion could result in cystic fibrosis. So I'm asking you to explain, how does taking out those 3 nucleotides affect the structure to result in this problem with cystic fibrosis? So think about what we've been saying about proteins. So that's the first one you're going to do and write down what your answer, what your thought is. Okay? Stop share. Okay, so that is it for our protein discussion. Hopefully that gives you a little more background and a little better understanding of proteins and how they work. We've got one more nucleic acids left to come.